Hi, my name is Soren Spina, and I'm a biochemistry major at Clemson University. I'm very excited to tell you guys about the human MUTS alpha DNA lesion recognition complex. This protein serves as the first step in DNA mismatch repair. Its inactivation is a main cause of hereditary non-polypsis colorectal cancer. The MUTS alpha complex is made up of two subunits, MSH2 and MSH6. Together, the heterodimer is capable of working in either the 3' or 5' direction. Now, for the first one that we'll show, the MSH2 subunit, possesses two flexible domains, which are able to remove a T from a GT mismatch without causing more harm to the DNA. Before this can occur, though, the MSH6 subunit, which we'll show in a minute, basically widens the groove in the mismatch, strategically forming a tilt. And this tilt allows the carbonyl group to interact with the guanine rather than the thymine. I like to think of it as almost distracting the DNA. Now, the DNA is initially bent over, and our protein's active site takes advantage of this. The heterodimer utilizes ATP hydrolysis to release mismatched DNA. Now, once bound, MUTS alpha can recruit MUTL alpha, which this protein then continues the repair, and it recruits it by binding ATP to MSH2. The release of DNA is triggered by ATP being binded simultaneously to both subunits. Now here we'll show ADP bound in ATPase sites. These sites are both basically superimposable when ADP is bound to them, and it's worth noting that the motifs known as Walker loops are completely ordered in MSH2, giving it a higher binding affinity than the non-ordered Walker loops of MSH6. The Walker loops are responsible for binding magnesium, which serves as a catalyst. With the absence of magnesium, there's an unbalance of charge that will impact DNA and ATP binding. Also, I find the dispersion of water molecules, shown in tan, very interesting. I believe it's as hydrolysis has already occurred, and that's why there's very few water molecules at the ADP side of the molecule. Here we can see negatively charged red DNA interacting with primarily blue positively charged sites on the protein. This is a better view of the GT mismatch, where you can see green guanine paired with blue thymine. Here are my references, and thank you very much to PyMOL and ChimeraX. Thank you guys very much. I'm Soren Spina, and if you guys have any questions, please feel free to contact me.